Today we will analyze SNPs using TRANSFAC, a little bit of machine learning and a glass of water. And we will learn how little SNPs can influence our ability to learn. Learn to learn. Have a look. Hello everybody, my name is Alexander Kell. And today we'll be using TRANSFAC to analyze SNP associated with educational attainment. Basically, we'll talk again about combinations of transcription factor binding sites in genomic enhancers marked by SNPs found associated with educational attainment. And that was done in a GWAS study of 3 million individuals. Basically, this paper. It was published recently in Nature Genetics this year, and the authors, they analyzed genomes of 3 million individuals, and they actually found these SNPs. You see, this is a Manhattan plot, so-called Manhattan plot, and in different chromosomes you see the peaks, and the peaks correspond to those SNPs which are found statistically significantly correlated or associated with educational attainment. And immediately a lot of discussion started in the press that actually our level of education is controlled by genes. The answer is no. Actually, the total influence of genetic variations is very small. It explains only about 4% of variations in educational attainment across those individuals which they checked. And actually, they also showed that educational attainment is primarily influenced by environmental factors or social factors, but they also influenced by some genes, and of course some of those genes are controlling cognitive functions like memory. And they also found that these genes are in fact influencing virtually to all stages of development of brain or development of neural communication. And basically they also found that all the genes or many genes near the lead SNPs were enriched for expression in the central nervous system. And we found the same thing. So we took those SNPs, which were around 850, which were reported in that paper, and mapped them to Gene Ontology. And as you can see here, we found enriched the following very interesting categories like regulation of neural differentiation, synaptic signaling or synapse organization, also neuron projection morphogenesis, and other very important functions which are relevant for brain development. But as many things in biology, there is another side of the coin. Actually, when we took those genes and mapped them to markers of human diseases, we found actually a lot of human diseases related to those genes. So you see here neurodegenerative disorders, schizophrenia, autism, and other disorders, which makes it a very interesting point that analyzing those SNPs is not only helping us to understand how our genetics helps us to sit calm in a class and learn, but also tells us about human diseases, and that's very interesting. So let's go back to our beloved transcription factors. Here they are, so these funny guys, and when the cell is developing or growing, there are many factors which are active in the cell. And as we know, they are bound to different places in genome, so-called regulatory regions of genes, either enhancers or proximal promoters. And what is interesting, they are binding in cooperative manner. What is it? It is important to understand the nature of this cooperative binding. Recently, it was shown that the cooperative binding of transcription factors is going on according to the so-called phase separation model, which you can see in the glass of water. Let's take a glass of water and put in it a little bit of oil. And you can see those droplets of the oil are quickly start binding to each other like transcription factors would do. And finally, those liquids are phase separated. In the nucleus of the living cell, this nucleation of the binding is actually promoted by the DNA sequence of the enhancer region where those transcription factors are actually attracted. And if this region has a mutation which actually destroys the binding of those transcription factors, then this nucleation is not happening and formation is dissolved. But what happens when there are many different transcription factors in the cell? So like in this picture which you see here, actually they found that it's the same thing. So those different transcription factors, they can organize together, they can bind together in form so-called condensates, which are either homogeneous or heterogeneous. So they can consist of several transcription factors acting together. And recently, it was demonstrated that those conglomerates, those concentrates of different transcription factors can be seen in the microscope. They actually can see them as a physically observed droplets in the cell. So the DNA binding domains allow transcription factors to bind to specific enhancers or promoter regions, promoting then the nucleation of these condensates of transcription factors as specific genomic loci. And that happens through so-called Idr Mediated Interactions. Idr here stands for intrinsically disordered regions which cannot form any stable structure by themselves. So, and they can only stabilize when several factors are brought together in close vicinity, and then they temporarily can stabilize the structure. And at the end, they form this condensate of many factors together. But the crucial point that these two factors have to be brought together to the close vicinity that they can interact to each other. And this is done by two sites in enhancer which are located in a close proximity to each other, and they can recruit those factors and bring them to the close vicinity to each other. So several sites in one region, so-called composite module, can attract several transcription factors which can now interact to each other and form this heterogeneous condensate. And then at the end, it leads to the high expression of that gene. If we have a SNP in that region, which is associated with that trait which we are analyzing, we assume that this SNP is actually marking this whole regulatory region of that gene. And each individual has a particular set of alleles, the haplotype, and in some cases, these alleles can change the binding sites for some of the transcription factors, these unhappy guys, as you can see on this picture. And then finally, these factors cannot bind to their places and they cannot interact with each other. And this condensate is not finally organized. This region is not prone for such organization of the condensate, and this leads to the low expression of that gene. So let's consider these three genes located somewhere in genome, and they actually carry some SNPs which are found associated with educational attainment. So basically those SNPs are located in the regulatory regions of those genes. And in one particular haplotype, for instance in this one, you can assume that these two SNPs, these two alleles, they are prone for binding of this regulatory complex and form this condensate of transcription factors, but one is not. But still these two genes are getting active, and it may happen that these two genes are actually control and they encode the proteins which are key proteins in this one particular pathway. So the pathway gets activated, which leads to the activation of the growth of neurons or glia cells. So we took the SNPs and put them into the GeneXplain platform, which you can see here. So, and then we use this part of the platform, which is called Genomic Variants. So we use different workflows and different methods from this part. Here is the list of SNPs which we downloaded from that website, and we uploaded them into GeneXplain platform. You see each line is one SNP. And then we map them into the genome, and then on this schema you see where those SNPs are located. So the SNPs are red bars, And in each gene, you can see the blue bars are corresponding to exons, and the R's between are corresponding to introns, and you can see also 5' region and 3' region of those genes. You can see how many SNPs actually were inside the gene. So basically inside the introns or exons of those genes, so including also 5' and 3' ends of those genes, so regulatory regions of the genes. And then when we analyzed those SNPs, as I explained in my lecture, basically we took the regions around the SNPs, pretty short region, 300 nucleotides around the SNP, and we were searching for enrichment of potential transcription factor binding sites. And here you see the results. So basically we found which motifs, which positional weight matrices from TRANSFAC were found to be enriched in those regions around the SNPs, in those 300 nucleotides around the SNPs. And here you see this list of the matrices, and here you see the motifs themselves from the TRANSFAC library. Next, we use these new methods called Miller, which finds the combinations of TFBS matrices. And this method uses sparse logistic regression to discriminate regions of our interest in genome from the background sequences. So in our case, we took these regions around the SNPs, and we took those matrices which were found enriched in those regions, and now this method tries to find combinations of those motifs which are enriched in these regions. This method was published a few years ago in that paper by members of our team. And it applies, as it was said, this sparse logistic regression. It takes the matrix, which is shown here in this picture, and scans it through the sequence. And if the sequence actually matches the matrix, then you get the highest PVM score. But we haven't put any cutoff for this matrix. We basically calculate the score in any position of the sequence, and as you can see here, there are some highly scoring sites and not so highly scoring sites. But then we sum up the scores across the sequence using this formula, and then using the logistic regression, we created a model which discriminates those regions from the background sequences. As a result of this Miller search, we found this list of motifs, which you see here. Combination of those motifs were found to be enriched in the regions around the SNPs. And here we present those found motifs with their weights, how they were included in the Miller model, and you see the top ones are SOX8 and several other matrices. When we took these factors and mapped them to the pathways, we found TGF-beta pathway is enriched, which you can see here on your screen. So basically you see those transcription factors which we found in that Miller model. At the top of the pathway you see TGF-beta, which signaling down to these transcription factors. And indeed in this paper it was shown that TGF-beta family signaling is very important for neural differentiation. For instance, during embryogenesis, TGF-beta family members regulate the initial formation of neural system and are involved in many other processes of neuron and glia differentiation. And here they also show how TGF-beta family members are involved in neural tube and patterning of spinal cord. Several SNPs were found near this TCF4 gene. And several of them are found actually inside the gene in the intron regions of that gene. For instance, this one is highly correlated with educational attainment, and we found actually 4 binding sites in the region around that SNP, as you can see here. So this region, 300 nucleotides around this SNP, which is highly correlated with educational attainment. P-value is very significant, and we found these 4 binding sites located in that area: SOX8, ELK1, FOX1 combination, and AV1 binding sites. The TCF4 gene is very important for brain development, and it's required for correct brain development in many, especially in the early stages of development. And disruption of that gene is actually leading to various diseases related to the compromised behavior, for instance, Pitt-Hopkins syndrome. And it was found that actually this gene is involved in development and maturation of various important neurons. And actually, when we look at this information about this gene in our database, we can see a lot of the biomarkers. And among them, you see schizophrenia, bipolar disorder, autistic disorder, and several other disorders which are associated with behavior. And this gene is actually expressed in many tissues, and it's quite highly expressed in brain. And as you can see here, this gene is involved in such important pathways, a VNT signaling pathway. And it's interesting to see that actually VNT pathway is connected to TGF-beta pathway, which we found also in this work. And especially this interplay between these two pathways is very important for stem cell self-renewal, and especially in neural stem cell expansion. And when we looked at the regulation of TCF4 gene, we see there are known sites in promoter of that gene for SOX4. And among the ChIP-seq fragments which we can map on the genome in the documented TRANSFAC database, we see that there are a number of regions for ELK1 factor binding and in the vicinity of that gene. And this correlates again with our finding that in that region around this SNP we found SOX8 and ELK1 sites, but we also found AV1 sites. And the role of those factors in the brain development is also known. For instance, SOX8 is well known as involved in neuron development, and it's known it's involved in neural crest development. As for ELK1, it's also well known transcription factor, which is playing a role in many facets in brain. As for the AV1, which is actually a proto-oncogene, it was found very early that it is required at mid-gestation for neural, heart, and paraxial mesenchyme development. In summary, we took SNPs associated with educational attainment. We put them on genome, and we found that many of them are actually located in regulatory regions of genes. And they're changing binding sites for transcription factors, which then destroy this complex organization of many factors together. As a result, those genes are changing their expression, which leads to different changes in different pathways related to development of brain, migration of neurons, synapse development, and other processes related to the brain development. To learn how those SNPs are related to our diseases, watch next video.